you everyone for being here today. I'll begin by doing an acknowledgement of country. I'd like to acknowledge the traditional custodians of the lands on which we gather today and pay my respects to the elders past and present. I extend my respect to all Indigenous people who are present here today in the seminar and who will be. I know not everyone is on as yet. And my respect extends to the First Nations and the Indigenous people around the world whose lands, stories, and wisdom continue to shape the fabric of our global community. So my name is Gail Cross. I'm an ID physician based here in Melbourne, Australia, and I worked as an ID physician and also in TB clinical trials when I lived and worked in Singapore. And it's my great pleasure to introduce Professor Gail Matthews.
Attention, please. Call adult.
I didn't think about that, did I? She is an infectious disease physician and program head of the Therapeutic Research and Vaccine Program at the Kirby Institute. She was recently appointed chair of the executive committee for the STRIVE Global Clinical Trials Network and was invited to facilitate to bring her perspective as a trialist outside of TB. Just a brief overview of things. We've got 3 speakers today, Professor Nick Paton, Dr. Maria Gler, and Professor Greg Fox. We will have time for questions for all our speakers at the end. We may have time for 1 question between speakers. I request that you keep your mics off. Videos are optional. And if you have any questions as they come up, I'd encourage you to put them in a chat. I've got Dr. Harry Walker, who's based at Western Health, isn't it, Harry? He's going to help me by looking through the questions and then bringing them up as needed. And I will begin by introducing Professor Nicholas Paton, who I have had the great pleasure of working with in Singapore. He is my PhD supervisor as well, and an overall top bloke. He's an ID physician and a clinical trialist, and his extensive work focusing on optimizing HIV and TB treatment in resource-limited settings. And this was recently recognized with an Officer of the Order of the British Empire. He has led the TRUNCATE-TB trial and is the core academic lead for the EU-funded UNITE4TB consortium as the Chief Investigator for its flagship trial, Paradigm for TB. So yeah, congratulations, Nick. I'll hand over to Nick and he can take the floor.
Well done, Sir Nick. Congratulations.
Nick, you're on mute.
Okay, the mute is dealt with. Now let's deal with the sharing slides. Gosh, it has so many windows open. Let's try that.
Did you get it from the King?
Not yet.
Oh.
In December, I picked up.
He's over in Australia at the moment, and it's interesting. It's not news at all. No one cares. Not one single state premier has agreed to meet him. Not one single. So we are getting towards being a republic, thank God.
Very good. Well, it'll last long enough for me to pick up my my goal. So that's.
Oh no, I'm sure he'll last in the UK for a little while longer.
Yeah. Anyway, good to see you, Mark, and hear you and many old friends I see on the call. So thanks very much, Gail, for the invitation to speak. I've got a number of slides. Actually, I haven't had a chance to time this. So Gail, if I'm going on too long, please just interrupt. It's fine. It can stop at any stage. So this is what I plan to talk about. As Gail mentioned, I'll orientate this around TRUNCATE-TB, but the aim is really to talk about doing trials in Asia and the challenges we faced, and I'll use TRUNCATE-TB examples for that. And I saw there's some questions, so a bit at the end on capacity building, if there's time. So first of all, for those of you who don't know what TRUNCATE-TB is about, just a couple of minutes, quick whiz through the design of the trial so the rest of it makes sense. So the idea behind the trial was that essentially what the current therapy is doing, 6-month standard treatment for TB, is over-treating the majority of people. Most people are cured with much shorter treatment. We've known that for many, many years. And so instead of over-treating the majority of people to prevent relapses in a minority. The idea of the trial was to flip that and treat people for a very short period of duration that will cure the majority, and then deal with the relapses in the minority. And to do that, we defined a strategy, called TRUNCATE strategy, 8 weeks of initial treatment, and then stop, possible extension 10 or 12 weeks if people weren't responding very well clinically, and then follow for 96 weeks, retreat, pick up early, retreat relapses. And then you need to look at the end. Obviously, there are more relapses in this intervention arm. That's part of the strategy. So you need to look at a longer term outcome, 96 weeks, primary outcome, unsatisfactory outcome, which was people alive, free from TB, off treatment. And if they meet that outcome, which is satisfactory outcome, then we can look at all of the secondary benefits of shorter treatment on quality of life, time on treatment, etc., etc. To do this at the time, this is sort of 10 years ago when we were planning this, we couldn't put our finger on a single regimen we thought would perform best at 8 weeks duration for initial treatment. So we had 4 experimental arms based around standard treatment with 2 modifications: high dose rifampicin, rifapentine, bedaquiline, and then added linezolid or clofazimine as a 5th drug. We also set it up as an adaptive trial design, so with a multi-arm, multi-stage IDMC reviewing interim data and making decisions about stopping underperforming arms. And then we also set it up in a way which I think is going to become increasingly relevant to de-risk. So started off with enrolling people with relatively low burden disease, mild to moderate disease, and only brought in people later in the protocol when the IDMC said it was okay, to expanding enrollment to people with more severe disease, high episode burden, and so on. It was intended as an Asian trial and was predominantly an Asian trial done in 18 sites, the majority in Asia. I'll talk about that a bit more in a moment. And here the trial was coordinated from Singapore, the team on the top left-hand corner, done in 5 countries. So our PIs, national PIs shown there in Indonesia, Alina, Philippines, Vince, Uganda, Christine, Thailand, Anjali, and then India, Rohit. And many, many people, a huge team involved in delivering this. The strategy worked in terms of being non-inferior on unsatisfactory outcome, W96, with a bedaquiline and linezolid containing 5-drug regimen, with a number of advantages. The main one being the average time on treatment was half what it was in the standard treatment arm. And now what we're moving into is looking at biomarkers. This is really the other half of the TRUNCATE-TB, is saying, okay, well, if we can define a strategy works at 8 weeks, how can we then refine that to make better decisions and potentially improve outcomes from the strategy and also improve the performance characteristics? So a lot of ongoing work now looking at biomarkers to refine the initial treatment duration, and also biomarkers to look at, can we improve the monitoring, like the frequency, can we come up with a blood test that would tell us when people are at such low risk of relapse that they could stop follow-up. So now on to the challenges of implementing this thing. So this shows the timeline. So we started this in 2012. Actually, the concept was first fleshed out in 2010, but the first grant application went in 2012, and it took us raising all necessary funding, doing all the preparation, which I'll talk about, 6 years before the first participant was enrolled, 2 years to enroll, 2 years to complete follow-up, and data cleaning and analysis after that, which is ongoing. Now, in terms of challenges, one of the major challenges was that this is a strategy trial looking at really the benefits and harms of over-treatment versus under-treatment. And that's much more complicated than a standard TB drug trial, conceptually as well as implementation, and has some safety risks that need to be addressed in the trial protocol. And one of the initial issues around this, of course, is that if you can't explain what you want to do, or people don't, there are too many questions, then don't buy into it. So in terms of fundraising, this was a challenge. And the other challenge was at the point of submitting grant applications, we didn't know what regimens we want to test. That was really part of the trial preparation, to sort all of that out, what drugs we could get access to and what regimens we wanted to do. Of course, this is now all pretty much part of the course. This is the Paradigm for TB trial, which I'm also involved with, which has got, I think, now up to 18 experimental regimens going into the trial, none of which were specified at the time that the money was raised in the grant application. So these sort of platform trials are now almost the norm, where you can build a trial and then say, well, we'll figure out what regimens we're going to use later. But for TRUNCATE-TB, this was kind of a bit pushing the envelope, at least in TB at that time, which is a challenge. We did find sort of consortium of funders who agreed to support this part. Mixed funding from the UK. The bulk funding came from MRC Work and DFID, and then some money from Singapore. All in all contributions about 15 million Singapore dollars. I think it's about the same in Australian dollars these days. The funding from the UK came with two conditions. One, so I've mentioned, decide on the regimens, but also procure the drugs, or demonstrate that we could procure the drugs, which sounded fine, but they gave us 12 months to do that, or else things would be withdrawn. And that actually was pretty hard, given the number of, once we finally settled on the regimens, the number of experimental drugs that we needed to procure within the grant funding. So we managed to do that by some donations, some purchasing. Bedaquiline at that time was really restricted and very hard to get hold of, certainly for drug-susceptible disease. Got major legal issues needed to be sorted out. But we eventually got donation or ability to purchase the drugs. The next issue was shipping those to 18 sites in 5 countries. This essentially was a full-time undertaking for somebody for a year to 18 months to fix, and then substantial amounts of time to maintain within an academic institution without the resources of pharma and without the budget of pharma to do. And Gail was involved in this as well and knows how painful this is. And then, you know, you can put the greatest plans in place, but this is just one of our shipments of standard drugs from one of the generic manufacturers got impounded. You can see ants there, you probably just about make it out. Temperature excursions, part of the course. This was ants, and there was a cockroach in this particular shipment that resulted in it being impounded. So all sorts of unforeseen challenges with that, which really can prove very difficult for trial. The next challenge in trial preparation was building support, again, because it's a controversial study, and many key opinion leaders in the field of TB, which is very conservative, were somewhat against this protocol, was perceived to be too radical. But one of the most important things, and I'd recommend anybody doing trials to do this, and I think it's increasingly done these days, is reaching out to the community, you know, representative communities. And there's a TB Community Advisory Board, Global TB CAB, which was spawned really by the HIV CAB, and I met with them very early on with some trepidation to pitch this study to them. And I think if they'd have had a downer on it, I think that I'd probably given up, because there was enough pushback with peers in the academic sector. But fortunately, the community got it, were really strongly supportive and encouraging, and that's really what carried this through to actually getting done. In terms of reassuring people, safety was an integral and a very prominent part of trial design. I've listed all of the things that we put there and justified why this was a safe thing to do. The other thing that I did for the first time with this trial, which I would now recommend everybody to do, and we don't always think about this. Obviously, you have to submit ethics applications, but actually looking at the protocol, especially with multinational trials, different languages and so on, and inserting an ethics section in the protocol, which is written as a case to ethics committees, which can be translated if necessary. And we reasoned this according to principles of Belmont, as though it was a case. It was 10-page ethics section in the protocol, And we had a very august ethicist working in Singapore at the time who I ran this by, and we discussed it to make sure it was sound. And despite everybody's, the doom mongers who said you'll never get this through a single ethics committee, we got it through 22 local ethics committees, 9 regulatory and national ethics approval agencies, with some needed a lot of discussion, but none of them turned it down. And I think there's, setting up the protocol with those arguments reasoned was very helpful for that purpose. Back to the sites. So in the sites in the original grant, China, India, Indonesia, Philippines, Vietnam, it was a pure Asian trial. When it came down to it, China and India and Vietnam and Myanmar, for various reasons, pulled out or just weren't able to proceed. So we ended up, of the original sites in the preparation, moving forward with Indonesia, Philippines, and then added Thailand. So during the trial conduct, this is how it looked after a year of the 2 years recruitment. About a quarter of the intended sample size from the sites in Philippines, Thailand, and Indonesia. And so we made a decision to expand the sites and stop some of these 4 experimental arms and control arms that we could get to the target sample size in at least 2. If the data monitoring committee didn't recommend stopping those arms, as it turned out they didn't, they said, no, it's all fine to continue. So at that time, we had these sites in Philippines, multiple sites in Philippines, Indonesia, Thailand. And the reason for doing it in Asia is partly because I thought, well, there's a different kind of disease, TB in Asia, and this strategy may be particularly suited. But it was also to counteract the sort of global trend. And I'm very fond of doing trials in Africa, great friends, and had a lot of success across many African countries. But there certainly was at that time, and continues to be, a dominance of African enrollment in large clinical trials. This is study 31, fantastic, the gold standard of TB trials that led to the licensing approval of the rifampicin moxifloxacin regimen. But only 12% of participants in study 31, ACG 53, 43, 56, I can't remember, sorry, ACTG. But the enrollment was from Vietnam, China, Thailand, and India, but only 12% of trial participants were from Asia. A gross mismatch with the burden of disease. And so we did want to keep it as an Asian predominant trial. So we added sites in India, but also added some in Africa, minority, because we wanted the generalizability as well. We also, as I alluded to, the IDMC was happy for all arms to continue. None at that time were underperforming. So in the end, the TSC stopped midway 2 of the arms. In fact, one of the regulatory agencies helped us with that choice by not accepting the new formulation of clofazimine that we wanted to use, which was actually the branded product. And with that, we managed to complete enrollment. Uganda came in at the end. India came in the last 2 weeks. And we made our target in 2 of the experimental arms. COVID, it came on just as we wrapped up recruitment. The 2 were linked, and that was, big across all of the countries. But fortunately, due to huge amounts of effort from the sites, they're absolutely magnificent. The loss to follow-up was tiny, less than 1%, and we kept most people under active follow-up. A few people transitioned into telephone follow-up for short periods of time, but really a magnificent effort from the sites. And he obviously helped manage that from the coordinating center in Singapore. I wanted to make a comment about monitoring, because I think that's really highly relevant for Asian clinical trials, multi-center, multinational. We did a lot of central monitoring, so we had a very good Singaporean data manager who programmed over 800 edit checks automated, plus very, very fastidious checking of data was coming in. That was essential for central. But in terms of on-site monitoring, we went with local CROs. Frankly, on a budget of $15 million, we could not afford IQVIA. or the like. So we went with local CROs, a mixture of academic and private sector, but none of the big ones. So academically, HIVNAT did the monitoring in Thailand. Uganda, we had individual local monitors, and the others we did in Philippines and Indonesia and India. We used professional CROs, but locally based. But the learning point here, which you kind of knew already, but it was accentuated or highlighted the importance of this with COVID, is it's critical to supplement local CRO monitoring with monitoring from the coordinating center to make sure the specific processes are being followed exactly, and really interrogate beyond the basic GCP. And here's one example. So the problem with COVID is that our Singapore team could not visit the sites to do the supplementary monitoring. And so we found things like this. At one site, we found that the entire process of recording results from one of the midget machines, and that was 1,000 samples, was erroneously recorded, and it was irregular. So we would just have no confidence in anything. And somebody had to go back to the machine data and the lab records and fix all of that manually, which meant that our 2-day or 3-day trip from the trial manager got extended to 2 months to fix that particular problem. And that's what goes wrong, or can go wrong, if you can't have detailed oversight. And then post-trial completion, and then I'll wrap up because I think I'm coming up to my time. One issue where I think is really important in these long-term major trials is the post-trial staff attrition. When you get to the end of the trial, you know, last patient, last visit, the money starts to dry up, and the interest starts to dry up, and people seek new or what are perceived to be more interesting. But actually, I think in the long term, the interest is when you've actually got the data and start figuring out answers, but people start to move on. And that's a real challenge. We found to be a real challenge. Cleaning data with unexpected surprises, not picked up in monitoring. I've given you an example of that. And then the statistical analysis. And let me just give you some examples of that. So one is statisticians, which are in short supply. And I think for capacity building, if we could build some Asian expert therapeutic area biostats expertise additional, that would be a huge boost. Anyway, in Singapore, there's this academic CRO called the SCRI, which we've worked with for a number of years. And there was a lead statistician there who'd worked with us on a number of previous TB trials. He was excellent, and he developed a therapeutic area expertise. He'd been involved in the simulations of TRUNCATE-TB from the beginning, done all of the interim analyses and drafted the analysis plan. And then 6 months before the last patient last visit, he moved to a private sector CRO, and then was out of reach and was extremely hard to replace in the academic sector. And so we had a process of trying to procure from our full service CRO and full cost CRO the services of the same statistician from the academic sector, which was challenging. Took us 9 months, and then he moved just as we got that contract signed to a new academic CRO. We had to start the whole thing over again. But fortunately, we've now resolved that. And it wasn't just the statistician, but it was also the data manager, the project leader, and many of the site staff. The other challenge post-trial is the sample management, especially if you plan a whole suite of biomarker work. So we've got samples concentrated in 2 repositories in the Philippines, just organizing the shipping and storage and monitoring of temperature control there is a huge undertaking. And the other one is in Indonesia because of the unique difficulties of exporting samples from Indonesia. And now we've been in the process for the last 2 years of getting samples and MTAs and shipments and all that stuff done out to, in most cases, central laboratories. In some cases, we have to do in-country analysis, for example, the PK here. So I'll wrap up. Here are some lessons learned, or we sort of half knew already, but just have been confirmed with TRUNCATE-TB, the challenges of doing multi-center trials in Asia. One is these days, you know, people interested in doing different for drug trials, doing a single regimen trial, those days are pretty much gone. And so you're going to be dealing with multiple investigational products, and that takes a long time to sort out, especially in the academic sector. Community engagement early to build a case of support, for many of these complicated trials these days is really important. Ethics, case in detail, the protocol has said. Sites, we kind of all know this, but essentially have a surplus. I would say if you're going to involve India in Asia, and I would say that's really important to at least try and do, you have to do that early on, but potentially can pay dividends later. Lastly, when you actually haven't had time to talk about that, we can perhaps cover that in the questions. Really, trials can be very powerful for capacity building, but need to supplement that. Monitoring on site, but need to supplement that locally, and then with an academic monitor, academic team monitor, and then plan resources for the post-trial, because that's can be really challenging. And again, just to put up, you know, I get the privilege of presenting this experience and these data, but again, it's a huge team, many names in small print, but people have, you know, made substantial amounts of time contribution. This is just the PIs at the sites. There's probably 200 or 300 people who deserve to be on this slide, and of course the participants who took the risk of enrolling in this, and the funders. Again, thanks very much. Happy to answer questions or discuss now or later. Thanks.
Thank you so much, Nick. That was fantastic. A real tour de force of the work you've done. I will open up the floor, but before I do, I think Professor Boyd had a question about the biomarkers that were embedded in TRUNCATE-TB. What were they?
So in terms of real time during the trial, we collected routine clinical data which are incredibly important for biomarkers as a base, namely GeneXpert and chest X-ray stuff, and archive the films and make sure we centrally got the CT, the cycle thresholds from the GeneXpert, which often aren't reported to the site. So we made sure we gathered those in real time as important clinical biomarkers. In terms of what we're doing post-trial now as a centralized exercise, RNA transcriptome, proteomics, using the Olink platform, 5,000 proteins. Obviously, we've done whole genome sequencing, so everything that was cultured post week 8, so from the time of completion of treatment and anything after that has been sequenced. Sputum test, we're doing sputum LAM with the Otsuka, originally Otsuka, now it's with another Japanese company, PHC, their test on archived sputum. Plate MIC testing for subthreshold MICs going into the trial, metabolomics, there are probably others, artificial intelligence on the chest X-rays. That's all I can remember, but if you've got any other ideas of add-ons, you're very welcome. We're at capacity. Gail,
go ahead.
Nick, thanks. Amazing effort. And I'm going to ask a question which is actually sort of serves my own purposes as well. I'm not a TB expert, but you know, running international, multi-center, multinational studies is a huge undertaking. And one of the things that we have constantly come up against is the sort of significant delays in getting regulatory approval through multiple countries, and the sort of extended time, and I can't remember in your timeline how long that took to get them through all the countries. And that's bad enough in a non-pandemic state, but when you're trying to do pandemic work or you know other sort of work that needs done quickly, it's a real problem. Did you have any experience? Have you thought anything about cross-regulatory approvals? Did you sort of try and go down that route of seeing whether you could get regulators to talk to each other in terms of their approvals, and sort of trying to hasten things through those processes, because they are incredibly delaying sometimes, aren't they?
Yeah, no, thanks, Gail. That's a really important point. And many of these countries do not have central national IRBs. They have central regulators, obviously. So that it extends this work. I guess we didn't, partly because it was such a bold design, at least for its time, that we felt we needed, you know, sort of individual input with the ethics and regulators, and many of them, you know, there was quite a bit of to and fro needed. But to your point, what we did do was try and get IRB approval in Singapore and Singapore regulatory approval, even though actually we didn't enroll any participants in Singapore, and really after a while realized that there was no point in enrolling in Singapore because actually the success would be much more in the region. But we made sure we got Singapore regulatory and IRB approval first, because many of the regional countries will look to robust, but actually all of these countries got robust processes now, but sometimes Singapore is perceived, at least for drug regulation, to be a regional leader. So I think that, and we pointed, each time we got an approval, we would feed back to the ethics committees and IRBs and the regulators. So I think that sort of helped build some momentum there. But at the end of the day, they all make their own decisions. The toughest nut came at the end. So despite multiple approvals from multiple IRBs and countries, India was the hardest one, and they really I personally had to go to the regulator meeting in Delhi on the third attempt to get this through, because the investigators had been beaten back on the first two occasions, and I just went and sat there, and it was really quite an ordeal, and they were listening to no one or nothing that had been done already, and they wanted to pull it apart from scratch.
Yeah, no, thanks. So it's something, if we could all try and, going forward, I think particularly as adaptive platforms are more recognized, hopefully streamlining some of those processes, I think it's a real need for global clinical trials ecosystems to try and do this.
Agree.
Gail, do you want to take any more questions now, or shall we move to our next speaker and save some for the end?
Yes, let's move on to our next speaker, please.
Okay. All right. So it is my pleasure to introduce, now, is it Maria or is it Maricel? Maria, I heard you.
It's okay.
I'll use the easier one. Dr. Maria Gler is an adult infectious disease specialist at Makati Medical Center in the Philippines, and she's been caring for patients with MDR-TB since 2005 and conducting MDR-TB clinical trials since 2008. And she was the lead author of a landmark trial on delamanid for MDR-TB treatment. She also leads the De La Salle clinical research site within the Emory-CDC Clinical Trials Unit, which has conducted trials on MDR-TB preventive therapy, treatment shortening studies, and also hepatitis B re-vaccination in patients with HIV. That's something else after my heart, but I will hand over to Maria, who's going to give us a talk now. Thanks.
I cannot unmute when I'm sharing, so I have to stop sharing.
Do you want to try again?
Yeah, here. Can you see my slides?
Yes. Do you want to, can you put it in presenter view maybe? There you go. Yeah.
Yeah. So thank you, Gail. And thank you, other Gail, for the invite. Thank you also, Dr. Paton, for referring me to the group. So for this morning, and good morning, for this morning, I will be speaking about the challenges and opportunities of implementing TB clinical trials in our setting. So the Philippines is a low and middle income country with a very dense population in most areas. The densest of which is Manila, Metro Manila, with 21,765 persons per kilometer. So the incidence of the prevalence for TB is 591,000 per 591 per 100,000 population, and TB is still the number 2 cause of death in the country. For MDR-TB. The prevalence is 20% amongst the previously treated and 1.8% among the new as of the last drug-resistant surveillance. So the health system is devolved in that healthcare is taken care of by the local government. But for TB, the national government provides treatment and diagnostics, and the local government provides staffing and infrastructure. So TB incidence has been increasing over the last 10 years, and the country also has an HIV epidemic that is the highest globally, and it has contributed to the increasing rate of TB, no? And MDR-TB, drug-resistant TB notification has also increased over the last 10 years with the wider access to rapid diagnostics, mainly GeneXpert, and GeneXpert as of availability as of 2024 January was provided. There were 1,022 GeneXperts all over the country, but this only covered 36% of the local health facilities. So in 2023, 9,800 patients with RR-TB was detected, but only 76% of these were initiated into treatment. So for our setting, our site is located in Cavite province. This is an area just south of Metro Manila, and we're specifically located in Silang, Cavite, a municipality with a population of 295,000, and the province belongs to Region 4A, which is the highest prevalence of TB in the country, second to Metro Manila. So for our site, for the whole province of Cavite, there are 30 MDR-TB treatment centers and 75 local health facilities, and the site has established a good relationship with the treatment centers and local health centers. So our site belongs to a network called ACTG, advancing. Before it used to be AIDS Clinical Trials Group, but they recently just changed the name to Advancing Clinical Therapeutics Globally for HIV, TB, and other comorbidities. So this network conducts research to improve management of HIV and its comorbidities, develop a cure for HIV, and innovate treatments for TB, hepatitis B, and other emerging infectious diseases. And it's funded under the National Institute of Allergy and Infectious Diseases and of the National Institutes of Health of the US, which means we are funded, we were given a core funding to run even without trials. No? Our site was identified as a protocol-specific site for Phoenix and MDR-TB preventive trial in 2016, and we were identified as a full-fledged CRS for a 7-year cycle in 2020. So over the years, we have developed the capacity to implement registration quality trials. And this is just a picture of our staff, the recent picture, because we started with a few, and this is now the numbers that we have. So this is just a summary of accruals of all our trials since we started enrolling in 2019. And the open trials are the A5300P or the MDR-TB preventive trial, which I mentioned earlier, or the Phoenix trial. And we are actually the highest enrollee globally in the ACTG. And the dramatic trial. This is a duration study for MDR-TB funded by DMID under the Boston University, and we're also the highest enrollee, and there are only 2 sites. So our retention rate is 98% across all trials. So for our challenges, number 1 challenge is coordinating with the national program and the local government units, because as I said earlier, for TB program, it's the national government national TB program, which provides drugs and diagnostics to local units. So for all clinical trials for TB, we need to get an endorsement of the national TB program, so we can approach the local units, the local health centers. And This takes some time because you need to complete your regulatory approvals first before you can ask for the endorsement, and the endorsement takes some time as well. So it adds up to your timeline. There's a difficulty in hiring medical and nursing staff who will be interested to work full-time in a research organization. So here, research career is not really that established. So doctors would want to go into clinical practice. Fortunately, we were able to hire 6 full-time medical officers, which chose to have a career in research. And capacity building for site and staff, since there was no site management organization in the country, we had to look to other countries. So we our staff trained in Thai HIVNAD and South Africa, and we also benchmark their practices and policies. So one of also the biggest challenge was introducing clinical trials to the community. But since we ACTG required setting up of a CAB, a community advisory board, so we were able to organize a community advisory board, had them trained by the global TBTC CAB and global ACTG CAB. So we now have a very strong community advisory board, which I'll show you their activities later. And we also have a high staff attrition because when you train people, CROs, either the CROs, which pays higher, or they would go abroad, would be getting them. So we had to come up with competitive salaries to be able to retain our staff. And of course, one of the also difficulty of the challenge of our staff is this encounters with the monitors, because we have a high enrollment, monitoring is almost monthly, and we encounter some aggressive monitors. So that was, we had to do a lot of conflict resolution for that. And we had conflicts with the initial institution that we were affiliated with regarding utilization of research funds. So we had to form our own entity. So there were a lot of opportunities rather than challenges, one of which is the partnership with the national program to somehow improve care. So for the MDR-TB preventive trial, since we are closely monitoring the household contacts, we were able to detect early incident TB among household contacts. So to date, among the 347 household contacts that we've enrolled, we've detected 13% of incident TB cases, and more than 80% of them were children. So the country's detection rate for children is only 9%. So amongst all the RRTB detected in 2023, only 9% were children. So because the contact tracing is only done at the outset, at the enrollment, and there's no follow-up. So we refer these patients to the local program for treatment, and we also relay our results to the national government to improve contact tracing activities. So another in the dramatic trial, which is an MDR-TB regimen duration trial, we have screened a total of 2,004 patients since 2022, and amongst the screen failed, 31% had FK resistance, because we would routinely do fluoroquinolone resistance for eligibility. And we were surprised to see that we had a high rate of fluoroquinolone resistance, and only 3 of these patients had previous exposure to fluoroquinolone. So the last DRS surveillance in 2018 showed only a 1% fluoroquinolone resistance. So we also relay these results to the national program. So for our community advisory boards, we have a very active community advisory board. They would have like monthly to bi-weekly community engagement activities. They would do TB and HIV awareness programs in communities, in schools, in job fairs, in hospitals or the industry. So our area is a factory zone or an industrial zone. So there's a lot of factories, and they would do HIV screening during these community engagements. They were also trained by the global CAB of ACTG for protocol review, for counseling. And it has actually made our collaborations with the local centers easy because of their intermediation. So just to show their the impact of their community engagements for 2024, for the first 3 quarters of 2024, they have already done 24 community engagement, reached almost 5,300 people, and screened for HIV almost 3,300 people. They have detected 77 persons who were HIV positive in these community engagements, and surprisingly, 12% of the 77 are females. The country's HIV prevalence is almost at 98% amongst men having sex with men. And yeah, this is the women's cases are probably underreported based on their findings. So one of our challenge also is to ensure that our young medical officers will develop a career in research. So we had to collaborate with the Emory-CDC TB Research Advancement Center for mentoring and grant writing, and 3 of our medical officers have had pilot grants on TB and undernutrition, VOT and MDR-TB, and a DCE for long-acting injectables with Gates MRI. So since we have a young staff, they would propose innovative strategies for retention. So we give our participants a birthday cake, give tokens if they were 100% compliant for the 6 months. So we actually have a more than 85% adherence rate across all our trials. And for the patients who have completed their treatment phase, they would have monthly activities such as poster making, TikTok video making, they would have a check-in virtual meetings monthly for those who have completed the treatment phase, and graduation activities if they have completed the follow-up phase. So in summary, establishing a clinical trial unit that can implement registration quality trials in a low and middle income country is feasible. Of course, with external investment, we had implementing TB clinical trials in high-budget country could improve TB programs and increase opportunities for young researchers. So that's it. And I will stop sharing. Thank
Maria,
you.
thank you for that wonderful talk. Really interesting to sort of hear from the sort of, that perspective of setting up that clinical trial infrastructure in a low middle income setting is really valuable. I think there are a couple of questions in the chat which we could probably just take before we move on to the next talk? If you're happy, Gail, should we do that?
Yeah, yes, please. Yeah.
Yes.
So one of the questions, oh, go ahead, Gail.
No, you
Yeah.
go. You go. You can read them very easily.
Maria, that was incredible. Thank you so much. One of the questions from Christine, how frequently did you do household contact tracing in your program? And a follow-up question also from Christine was, did all patients use internet-based communications in the trials?
Okay, so for the program, the national program does contact tracing, routine contact tracing on enrollment for MDR-TB patients, but for drug-susceptible TB patient, the drug-susceptible TB patients, there is no established or there is no routine contact tracing. It has been in place when the MDR-TB program, which is a very recent program, was established. So it's better in the MDR-TB program, but it's only done one time at screening or at initiation of treatment. So there is no follow-up. And with with the clinical trial that we are doing, where we observe the contacts for 96 weeks, you would observe the cases or the incident cases like from 6 to 12 months on observation. And this is with intervention, no? And how do you manage side effects for DRTB? So it's one long lecture, so probably. So, but our staff are trained for the management of DRTB adverse events in a clinical trial setting.
Sorry, go on. Sorry, Dr. Maria.
And the internet-based communication. So rich for internet reach, because we are in the peri-urban area. So the internet reach is very high. It's 80%. So we actually do VOT for all video observed therapy for all our patients enrolled in the clinical trials for MDR-TB regimen, not for the
Not drug susceptible.
Even for drug susceptible TB clinical trial, we do the VOT. But for the preventive trial, we're just doing drug accountability check.
Are there any other questions?
I think that was just a clarification also from the person who asked that question. When you collect the side effects, are you collecting them online or on a paper-based?
Oh, it's paper-based, because they visit us often during the treatment phase, so we do paper-based adverse event collection.
I might take the opportunity to ask a question of Dr. Gler. One of the things we've encountered is sometimes when you go through the effort of running a well-conducted clinical trial, you get good data, and you feed that data back to governance bodies, TB programs, for example, in country. The uptake of that isn't necessarily what you'd hope for. And I wondered if you've encountered instances in your experience where that's happened, or generally because you've got the benefit of already engaging the TB, if I understood correctly, the National TB Program, and you've got such a robust CAB, whether you haven't encountered that because you've already got so much backing for any data that's produced within the Philippines.
So the country program has already been since they had wanted to be involved at the outset, like at the start of the trial. So they already know what trials are ongoing, and we give them our promise to them was we give them a quarterly update on enrollment side effects. So we have a quarterly meeting with the national program to give them an update on our activities, on our trial activities. So they already are fed with the data during the trial implementation.
Okay. And they take, as in that results in changing policy in how TB is treated as well in the Philippines.
So it has actually changed policy on the treatment of MDR-TB in children. So we were doing the dose ranging study for delamanid in children. It was very difficult to encourage them that children need to be treated based on clinical diagnosis as well as bacteriologic diagnosis. It was a long hurdle, but now treatment for MDR-TB in children is already increasing in the country. It's still far from ideal, but
yeah, but small increments, hopefully. I think we are at time, Dr. Matthews. We might move on to our next speaker.
Absolutely, and people can think of more questions. I think there'll be more time for questions at the end so that people can be thinking during, well, and listening to Greg's talk. So it's my pleasure to introduce the final speakers, Professor Greg Fox. He's the Interim Director of the Sydney Southeast Asia Centre at the University of Sydney and the Professor of Respiratory Medicine there. And for over a decade, he's been leading clinical trials and research on infectious and chronic diseases in Vietnam, particularly focusing on tuberculosis and lung disease prevention. And his work has influenced global TB policies through research and WHO guideline development. Clinically, he's also serving as a respiratory physician at Sydney's Royal Prince Alfred Hospital and the area director for tuberculosis services. And is dedicated to building research capacity both in Australia and internationally. So thanks, Greg.
Thanks very much, Gail, and thanks everybody for joining today. I have about 20 minutes, and what I hope to do in that time is to I think take a complementary approach to the other two speakers. I'm going to be talking about some of the pragmatic trials that we've done in Vietnam and try to combine an exploration of the findings of those trials and how they have been focused on policy and practice, but also some of the lessons that we've learned through doing those trials about the development, implementation, and the translation of our trial research as well. Just as a declaration, you can see here that we've had some support from the pharmaceutical industry, but this is not directly relevant to what I'll talk about today. So just to take a step back from the discussion today, and just to remind everybody online that the reason that we do trials in the Asia-Pacific region is because this is really the ground zero of the tuberculosis pandemic worldwide. That is, over 60% of all cases of TB occur in the region. And as Nick said and illustrated so well, it's vital that we do trials in these populations so that we can understand what works and that we can make sure that the guidelines that we develop are relevant to those populations. You can see here that in the Western Pacific region, of which Vietnam is a part, that there has been a slow decline in tuberculosis over the last decade or so. But really, in contrast to the End TB strategy, we have not seen the dramatic reduction that we need in order to achieve TB elimination. And so for this reason, when we're designing our clinical trials, we need to be thinking about what it is, what is the evidence that we lack that's stopping us from making this major progress. Vietnam, which is the country in which the research that I'll present today has been done, is a middle-income country in Southeast Asia, a population of around 100 million people, and it is one of the high-burden settings for both TB and for drug-resistant tuberculosis. It has a nationally coordinated TB program led from the National Lung Hospital in Hanoi, and it has, over the last decade and a half, been involved in many clinical trials from many large international groups and collaborations that you would be familiar with. Specifically in relation to the work that we do in Vietnam, Our work has been done through Australian research institutes. The Woolcock Institute implemented the 2 trials that I present today, and now we've transitioned to the Sydney Vietnam Institute, which is a University of Sydney research institute based in Hanoi and Ho Chi Minh City that has clinical trials capacity. And we work very closely with the National Tuberculosis Program. And just before I begin talking about the particular trials, just to acknowledge the National TB Program, and particularly their director, Dr. Luc, and their leadership team for support over many years. Now, in relation to the way that we have thought about the trials that we do in Vietnam, really you could break down the trials into the broader public health objectives that we have, that is to detect TB, to prevent TB, and to treat TB. And although there's obviously a lot of detail in each particular trial, I think it's really helpful to try and think about how the trials that we're doing fit within the context of the broader goal for TB elimination. That is, in the case of detection, that we're aiming to try and reduce that gap between cases that are detected and cases that emerge. In prevention, to think about how we can stop new cases from developing, so to turn off the tap of transmission. And then treatment, how to quickly put people on safe and effective treatment that can result in better outcomes for individuals, but more importantly, that can stop the pandemic from spreading and perpetuating the transmission in the community. I'm going to talk about 2 studies today, the ACT2 trial and the VQuin trial, which are both trials done in Vietnam to answer particular questions. And I'll explain as I go the reason I've chosen those 2. Just to make a point, as with the previous speaker, we're also a part of a number of trials networks. And I think, one general comment would be that there's a real benefit, I think, from being a part of these large consortia. The ACTG, you've heard about already. The TB Trials Consortium is another one, which is based out of the US CDC, that implemented the Study 31 that Nick mentioned. And then CABV, which is a Canadian led from Canada, Australia, Benin, and Vietnam, which we're a part of as well. And I think for people who are online who are interested in getting involved in trials for the first time, I'd really encourage you to try and get involved in existing trials consortia, because they provide opportunities not only to learn about trial design and trial methods, but also, as you've heard from both of our speakers so far today, there's great opportunities for mentorship and growing capacity for people from LMICs as well as from high-income countries like Australia. So I'm going to talk a little bit about pragmatic trials. We've heard today in the form of TRUNCATE-TB and the Phoenix study discussion of phase 3 trials, which are implemented with regulatory focus. But I think it's important to remember that clinical trials are not always for the purposes of regulatory approvals of drugs. And in fact, many of the gaps that we have in evidence actually are gaps in interventions that may not be drug related. So in the case of tuberculosis, active case finding was an area where there was very little evidence for a long time based upon high quality studies. Now, the problem that we were seeking to address in the contact tracing studies and the community studies that we've done is this global TB detection gap. And it's estimated that there may be around 3 million people a year that are not detected. And again, the epicenter of that problem is in the Asia Pacific region. And so for that reason, we, about 15 years ago now, worked with Professor Nhung, who was the director of the National TB program, to develop a trial that could answer this question. And this is a particular type of trial, which is called a cluster randomized trial. Now, individual randomization is what we're probably most familiar with, where we randomize an individual person to an intervention or a comparator. But in the case of health system interventions, randomizing the whole health facility can be a more pragmatic way of implementing the study, because it is easy to train everybody in the facility to do one thing rather than for them to have to change back and forth. And particularly when it's a pragmatic study, where you're looking to implement it within routine care without supplementing it with a lot of additional staff, this approach can be quite effective. So the ACT2 trial was conducted in 70 different hospitals around Vietnam. You can see here the arrows showing the different provinces that participated. Yellow was the intervention and green was the control. And the reason that we did that was because it was a very pragmatic trial that was designed to be implemented within a whole health facility. This reduces a contamination effect between groups. So the ACT2 trial really sought to answer this question: how can we increase case detection amongst people who are household contacts of patients with just newly diagnosed TB? It might seem like a question that was obvious to answer, but there had never been a clinical trial to demonstrate whether this actually was effective or whether people would have come in eventually with their TB for diagnosis. And so the primary objective you can see here is to work out what is the cumulative case detection rate of contacts for TB amongst household contacts of patients with smear-positive TB. And we had a number of secondary objectives that we looked at, including feasibility and cost effectiveness. And this particular study was very pragmatic. We just enrolled everybody who was eligible to be treated through the Vietnam National TB program at the time, so smear-positive adults. And we then enrolled all of their household contacts of anybody of any age who'd been living with them in the household in the previous 2 months and who hadn't been taking treatment. And again, being very pragmatic, we had the intervention groups where we conducted screening using existing tools. So it was chest X-ray, which was read by the local doctors with a second reading by a provincial doctor, and then symptom screening combined. And then we screened them 4 times over 2 years at 0, 6, 12, and 24 months. And the comparator group was just usual review at 24 months. So one point to raise here is when you're doing these pragmatic trials, one of the ethical issues that we face is whether it is ethical to provide no intervention in the control groups. And in fact, at this particular time, which was in the 2010s, WHO only had conditional recommendations for contact investigation, and contact investigation was not done routinely in Vietnam. And so after discussion with our ethics committee here and with our colleagues in Vietnam, we decided that it was ethical to have observation without screening. You couldn't do this trial now because there's now strong WHO recommendations that have arisen as a result of the work that you'll see now. So the sites are shown here, and I won't go through the detailed outcomes, but essentially we enrolled about 25,000 household contacts across these clinics, and we used a very pragmatic approach. That is, we had paper register books which matched with the style of documentation that was used and is still used at the time. And we enrolled people and collected the data and entered it into a database which we developed. We had a study team that was based out of Hanoi. At the peak, we had about 8 people who were overseeing this trial, and that included a data manager as well as staff who would do monitoring, evaluation, and training across all these sites. The study was obviously a lot more straightforward than doing a drug trial because the risks of contact investigation were pretty minimal. We did, of course, want to avoid doing chest X-rays on pregnant women, but otherwise there was really very minimal risk in this particular study. So the primary results of this study, shown here up to 900 days post randomization or post enrollment, was that we had about 15,000 people in the control group and about 10,000 in the intervention group. And amongst the control group, the number of people notified was 88, which is a case detection rate of about 560 per 100,000, compared to 163 in the intervention group, which has a case detection rate of over 1,600 per 100,000. So that is, this intervention more than doubled the case detection within the national TB program of tuberculosis. And in fact, we've done subsequent work which follows people up even further, and there is continuing benefit in terms of case detection. We also, although I won't show it here, showed that there was actually, in post hoc analyses, there was a reduction in mortality that was associated with this intervention as well. So to say that this study has been, a large study, obviously is clear, but it's an example of a study which has also had policy impacts. So obviously, when we do clinical trials, we want to try and change the way that the problems are managed, not just in one country, but in other countries as well. And so as a result of this trial, the data were presented at a number of WHO meetings, and the strength of the recommendations for contact tracing was increased to a strong recommendation for contact investigation. And now in Vietnam, it's become a standard part of their national policy and an important priority as well. So I haven't presented the data here, but it was also shown to be cost effective. So this was a very pragmatic trial, not a drug trial, but I hope you can see that it's one that has significant policy implications. And I think as clinical trialists, we can often be looking at these sorts of health system type interventions to make big differences to the way that patients are cared for as well. The second trial that I'm going to talk about is the VQuIN trial. which has been completed recently and is currently in press. So this is a trial looking at prevention of drug-resistant tuberculosis. The context of this trial is that there has been a major individual patient and health system costs associated with drug-resistant tuberculosis for many years, that over 400,000 people around the world are affected by it. And prevention is a strategy which can potentially reduce this burden. There had been observational studies performed previously that suggested that preventive regimens were effective, but they hadn't previously been shown in clinical trial data to be effective. So the VQuin trial was a phase 3 trial implemented in Vietnam, and the objective is shown here: to evaluate the effectiveness of levofloxacin, which is a fluoroquinolone antibiotic, given for 6 months compared to placebo in the prevention of active TB amongst household contacts of patients with RIF-resistant or MDR-TB who have latent TB infection. We had a number of secondary objectives relating to safety and tolerability, mortality, adherence, and cost effectiveness, as well as acquired resistance. This figure shows the scheme of the study. So 6 months of levofloxacin or an identical placebo were given, and then people were followed up for a total of 30 months after the time of randomization. This was an individual randomized trial, although people were in the same household within 90 days enrolled of each other, then they would be given the same treatment to avoid a contamination effect. So this study is an investigator-initiated study. So in contrast to the big trials like the ACTG trials or TRUNCATE-TB, this trial was funded by just one funding agency in Australia, the National Health and Medical Research Council. And so therefore it was feasible for us to do this in one country, in Vietnam, but not in other settings. And I'll tell you in a minute about a sister study which also was implemented at the same time in South Africa, which is called the TB CHAMP study, which was also looking at the same drug, levofloxacin, but in kids. And because of the timing of these two trials, we had close collaboration with them throughout the design phase and also the implementation phase, which was proved to be quite important in the way that we interpreted our trials, as you'll see in a second. So the study population for this trial is patients who are of all ages, who are positive for tuberculin skin test or TST conversion, and who don't have active tuberculosis. There are some more detailed exclusion criteria shown here as well. So these are household contacts of index patients with RIF-resistant or multi-drug resistant TB. And so therefore they had quite a high risk of developing TB themselves. The way that we analysed the data in the primary analysis was that we looked at the incident rate ratio up to 30 months, and we had a secondary analysis looking at safety up to 21 days post-treatment. Now, as we were completing follow-up in this study, we observed a lower number of events than we had expected. This is before we unblinded the study. And so as a result of that, we talked with Annika Hessling and the leadership team of the TB CHAMP study, and they had also observed in their own study of kids that there was a lower than expected event rate of incident cases of TB. And so we pre-planned a secondary Bayesian analysis and a combined meta-analysis that combined results from both studies. A Bayesian analysis allows you to borrow information from one trial and apply it to the other, and it potentially increases power compared to standalone analyses. The TB CHAMP's primary outcome was up to 54 weeks, and so that was the primary outcome that was chosen for our analyses. And these analyses were done by Trinh Duong and others at the University College London. So it was a great example of collaboration between trials groups. So one of the messages that I'd like to communicate today is that it's hugely beneficial for us to be working together with other trials groups rather than just in silos. And I think this particular trial is a good illustration of that. The consort diagram here shows that we enrolled 4,100 household contacts for eligibility, and 2,000 were randomized, about a half to levo and placebo each. And you can see here that we had very high follow-up rates. So in the group that completed, there was a 98% follow-up rate, or over 98% follow-up rate, and slightly less in the group that didn't complete. But overall, we had about a 98% follow-up rate in this study, and we had very low rates of mortality during the follow-up period as well. You'll see here that we had 61 people who had co-prevalent TB. They were ruled out from being randomized, of which most of them were bacteriologically confirmed. And that number 61 will become important in a minute. This is the characteristics of the population enrolled in this study. You can see here we had few children. And the reason is that this is another ethical question. In the initial time at which we proposed this study to the ethics committee in Vietnam, they expressed concern around the safety of levofloxacin in kids. This is despite the fact that there was another trial underway that had already got pretty good data about safety in kids. And so as a result, they deferred approval for us to include kids under 15 until after we had provided them further safety data. And so just within the last 6 months of recruitment, we were able to provide that data, which was shared by collaborators from the TB CHAMP study, and then we're able to recruit younger kids. But as a result, the TB CHAMP study and our study were quite complementary. They had young kids mostly and some adolescents, and we had some adolescents and adults. Just some other features here in Vietnam, there's very low prevalence of HIV amongst people who are household contacts, just 0.4% had a history of HIV, and about 90% of people were either TST positive or had a TST conversion. So these were clearly a high risk group. This is a figure showing the Kaplan-Meier curve up to 30 months. You can see in the red, the placebo group, and in the blue, the intervention group. And so you can see that there's a separation in the first 6 months between the lines. But then in the final period, there was, in fact, 3 out of the 6 incident cases that were detected occurred at 30 months when we used sputum gene expert in addition to chest X-ray to try and rule out any early tuberculosis in this group. And so you can see that whilst there was a difference between the lines, and the point estimate in the primary analysis was 0.55, that is a 45% reduction, that because of the low event numbers, there was quite wide confidence limits. So you can see that there were 6 cases in the levofloxacin group and 11 cases in the placebo group. And most of those were bacteriologically confirmed, just a small number of clinically confirmed TB. In terms of the per protocol analysis, we saw a pretty similar point estimate of 0.6 with, again, wide confidence limits. Now, I mentioned before the TB CHAMP study. So the TB CHAMP study faced a similar challenge to us. They had pretty low numbers. They had around 1,000 people in their child study. And so they had 5 children who were diagnosed with TB in their levofloxacin group and 12 in their placebo group. And so overall, when we combined our studies together, we had a large number of 8 against 41. And so when we combine the analyses, you can see here that if we use the Bayesian methods that we had specified, sorry, this is not the Bayesian one, the next one is. If we use the individual patient data meta-analysis and combine the 2 studies together, as we had pre-specified, then you actually get a point estimate of 0.4, or 60% reduction, which now becomes statistically significant. And so you can see for both trials, we had the same problem of underpowering. And so therefore, the pre-planned analysis allowed us to address that. The Bayesian analysis that I mentioned before, when you apply the TB CHAMP findings to Viquin, you get a 0.41 hazard ratio. So again, with the credible intervals not crossing the null. And in terms of the safety outcomes, you can see here that the safety profile in grade 3 to 4 adverse events was no different between groups, but there was substantially more people, that 18 to 19% more people who had grade 1 or 2 adverse events compared to grade in the levofloxacin compared to the placebo. So there was low level intolerance there. And the same was seen in terms of the ones who stopped taking the study drug. So about 6% more people stopped taking levofloxacin compared to placebo due to an adverse event, which was pretty much grade 1 or 2 adverse events. And these were primarily intolerance, such as gastrointestinal intolerance, or some non-specific musculoskeletal symptoms. But we didn't see any evidence of hepatotoxicity or of tendinopathy between groups. And finally, the analysis also looked at acquired drug resistance, and we found no evidence of acquired drug resistance. We did whole genome sequencing comparing the index contact pairs, and we found that in 3 out of the 8 cases where we had paired data available, the incident cases had not been affected by their enrolled index case. So this is really interesting. it shows you that transmission in Vietnam of MDR-TB, even though it's common in the household, it's still driven a lot by the community transmission as well. And we did a number of other additional sub-studies, which I won't have time to go into today, but they included looking at the human microbiome effects of the drug, looking at health economics, and looking at biomarkers associated with TB treatment response as well. So in conclusion, the VQuIN trial showed that levofloxacin was associated with about a 45% reduction in confirmed TB, that there was a low incidence of adverse events, and that there was no acquired resistance. And then when we had pre-specified analyses, we were able to identify a consistent and significant effect. So just in summary, there are, I think, some challenges that we can see in the way that we implement these trials. And one of them, which I think is a major challenge for TB prevention studies, is that I think there are often a lot fewer outcomes than we expect. And so it's very challenging when you rule out all of the prevalent TB so carefully with sensitive tests to have enough events. And so these trials therefore have to have huge sample sizes, and hence multi-site studies are important. I won't go into detail, but growing our clinical trials team and also growing capacity in the TB program was a big part of the work here. And so as we've been establishing our team, we've also been working very closely with the local doctors and building capacity at sites as well. And there's also significant laboratory quality control challenges as well, which we can address, through various quality control measures as well. So just to acknowledge many of the people who contributed to these two trials who are shown here, particularly Professor Nguyen, the director of the TB program at the time, and also Professor Tuang and Bing, who was the trial coordinator for the VQuIN study, and then some people who are listed here as well. I'll stop there and happy to take questions. Thanks.
Thanks, Greg. That was a great talk, and it's so important to do work that obviously hopefully results in changing practice. And implementation work is obviously just as important as the drug sort of treatment trials. So I had a quick question whilst waiting for other things to come in. And it's for all the speakers as well. So in your trial, obviously levofloxacin was shown to be beneficial. So I presume you're hoping that it will translate into a change in practice or a practice recommendation. You excluded pregnant women. And I can understand why, but clearly there is a move to study more pregnant women within trials, and I don't know the TB field and how often pregnant women are included in TB trials. So, so really, I guess just a comment about then whether this would be translated into pregnant women and whether pregnant women should be studied in these kind of trials to gather data on what's important for their outcomes.
Yeah, thanks, Gail. I think that's a really important point. So to answer your first point, so in relation to policy, so WHO Guideline Development Group met last December, and in February of this year, levofloxacin is now recommended for all contacts of patients with MDR-TB, and the formal guidelines on the left there were released in September. So this trial has changed policy in combination with the TB CHAMP study. In relation to your question about the inclusion of pregnant women, so certainly there is, I think, recognition of the importance of including all populations who might benefit from treatments in the trials that we do, and I think there's really, you need to consider it on a case-by-case basis. And certainly we push to include kids despite the challenge from the ethics committee for the same reason. In relation to pregnant women, we did have quite a debate about it. We concluded that the number of pregnant women we'd likely to enroll would be so small that we probably couldn't learn anything meaningful about it, and that we thought that it was unlikely in this cultural context that people would want to take preventative therapy with levofloxacin. And also our practice in Australia, at least, is we very seldom recommend preventive therapy to pregnant women. We mostly wait till they deliver and then offer the pregnant women treatment. So in this case, we decided that the knowledge that we would gain would be probably not justified. But certainly in another trial that we're doing now, which is an isoniazid resistant TB, active TB treatment trial, we are enrolling pregnant women for that reason, despite the fact that that levofloxacin is in the regimen, because we think that the benefits of a woman with active TB of having a levofloxacin would be greater.
I might quickly take the opportunity to just comment, and perhaps if Phil Ducrot is online, give him the opportunity to speak towards this. Last year, we gathered a group of clinical trialists throughout the region and asked them to comment on what they thought were the important questions. And one of them in the TB clinical trial space, and one of them was really the inclusion of groups that had been excluded to date in trials. And Gail, yeah, we don't have, we certainly don't have as much data as we need to in a variety of populations. And that's because of always trying to balance, you know, getting good quality data from a very specific population and also the safety issues that arise with that. So I'm not sure, Phil, if you wanted to say any more about that and also ask your question that you've posted on.
Thanks, Gail. So I've popped the article you mentioned, which was a Delphi process of consensus on improving clinical trials. And there's a whole number of issues, and pregnancy is covered. I think the TB field has been quite conservative. It's learning and moving forward. I think we still find ethics review boards in many countries are very conservative when it comes to pregnancy inclusion. And so I think Nick's point on solid documentation of the ethical issues in the protocol is advisable and a key lesson. I won't talk too much more about that. But maybe just to ask my question, I think Greg and also Nick sort of alluded towards some, issues within the region. Obviously, the region has a lot of TB cases. But do you think we have specific issues that need specific regional focused trials? Or do you think it's really about just better collaborations, better regional representation to make sure that the sub-studies can pick up the regional differences? What's the way forward? What do we need to do differently?
I can start. Yeah, look, I think there are going to be some regional priorities, particularly around vulnerable populations in the region. And so some of the pragmatic trials we do, I think, we should certainly be looking to try and include people from Southeast Asia and from the Asia Pacific region. I do think, though, to change global guidelines, the more we can be including people from multiple regions, the more generalizable our results will be. So it's probably not a correct answer there, but I completely agree with the premise of your question that we need to be trying to enroll as much as possible people from affected communities. And since Asia Pacific is the home of TB globally now, we should certainly be doing that as much as possible.
Maybe can I?
Yeah.
I mean, I agree in principle. Of course, we're much better off with doing globally representative trials. And just as I alluded to in the talk, this tendency to split, or the dominance of African enrollment. One way that's played out. We've just been recently looking at the low-grade toxicity, and I was interested, Greg, in your talk where you emphasised that when you look at high-dose rifampicin and rifapentin, you don't see much difference from standard treatment at grade 3 or 4 level, right? But when you start looking at the low-grade gastrointestinal toxicity reports, it's a very stark difference, and that didn't come across at all in study 31, which may be, the fact that the enrollment was very much outside of Asia, or it may be actually that there's a difference in threshold of tolerability within Asia that may be genetically related. So I think it's really important to have representation of both of the major regions, ideally globally, similar to the trials that the Kirby does, somewhat uniquely global representation. I think that really adds a strength to it. The problem is often is the funding agency. So, for example, you've got the major European funding agency, EDCTP, which will not fund, unless they've changed, will not fund trials in Asia. So they've in a sense force Africa only European collaboration trials, which is fine if you compare that trial with a similarly designed trial in Asia. But this sort of create that there's a tendency for funders to create these silos as well. Yeah.
I see a
But I
couple
think
of people have turned their videos on. If you have a comment, please feel free to ask. Steve, do you want to ask a question? Steve Graham.
Gail, no, sorry, I actually lost connection for a while, so I've just come back in. So
no worries, that's fine. Catherine Berry points out that there are different lineages relating to TB lineages, which are different in Asia. I wanted to make a point. I do believe that there are potentially pharmacogenomic differences in how drugs are metabolized that I don't think we nearly know enough about in populations within Asia Pacific. And of course, acknowledging that that's really not one population, it's a huge diversity of population as well. Harry?
Yeah, I had a question, particularly for Nick Paton, but probably for all of you experts. We've all seen the success of SNAP, so the Staph aureus Network Adaptive Platform trial, and I couldn't help but think of a similar thing when you described how onerous it was to get this trial infrastructure and teams up and running, and then recruited for 2 years and it all stopped. So do you think there's a future for more indefinite, longer lasting network adaptive platforms in TB clinical trials? And then what are the barriers? What would be the issues with that?
Yeah, thanks, Harry. I mean, the main barrier is, of course, funding. Is finding a funder that really would commit long term, and many of them, even if they wanted to, can't, because they have funding cycles that they have to obey. So I mean, you know, with TRUNCATE-TB, we essentially set up a platform. You know, the MAMS trial design, you can add arms in or you can drop them, and it was based on you know, the whole idea came from colleagues at the MRC Trials Unit where I was working at the time, working prostate cancer, where I think the trial is possibly still going after about 15 years, where each time a new drug for prostate cancer comes along, they introduce that into the trial. So you know, it could have been maintained, but I mean, it's partly because we were totally exhausted by the end of it. Doing this on a shoestring makes you even more tired than it would be if you did it with, you know, a budget of hundreds of millions of dollars. But equally, you know, the funding was for a finite period, and you know, we had to close the trial down. Looking at, you know, other experience at the moment, so the EU has funded through the UNITE4TB network a platform trial, which is the one I'm involved with, Paradigm for TB, which I think in the initial wave has got 12 different regimens. We're just in the process of adding another 9 with some other novel drugs that are coming along. But you know, that's a 7-year funding span, which has got another 3 years to work on it, and we have to start thinking now about continuity of that post trial. I mean, it will be essentially the platform for TB phase 2, B and C before you get to phase 3 trials. Gates have funded pan-TB. They don't, you know, they have I guess an advantage of, in theory, indefinite funding, but they have milestones and deliverables to make as well, which can result in easy termination of that. So yes, in principle, but we need funders who would say, yes, you know, we'll fund this for 20 years.
Thanks.
I think my colleague Debbie Marriott from St Vincent's is also put in the chat just a plug for TDM. I think Nick might have mentioned, do the trials generally contain TDM sub-studies within them, or?
Yeah, so I completely agree with Debbie. You know, the PK is an essential part of interpreting these trials. So, you know, certainly for a treatment trial, I think it's not appropriate, unless it's a completely fully understood regimen. It's not appropriate to do a clinical trial these days without nested PK for novel drug regimens. So certainly in TRUNCATE-TB, we built that in, not in real time, because then you lose generalizability of your intervention. What you're trying to do in the trials, do stuff that's definitely replicable, especially, although it perhaps didn't come across from the way I presented TRUNCATE-TB, the thinking behind that was very much something that, how would we translate that into a program setting? And, you know, a lot of the outcomes, which I didn't talk about in this presentation, were orientated around participant perceptions and what programs would think and that sort of thing. So in the real time in a trial, you have to be careful not to do stuff which then essentially renders your trial intervention redundant, because that kind of monitoring wouldn't be possible at an implementation stage. But certainly, doing nested PK, I think, is vital. And you know, if you find stuff through that, for example, if we were able to find a threshold of rifampicin exposure that would strike the right balance between efficacy and tolerability, and I don't think we're there yet. But certainly, when we're going to have a good go at that with the PK analyses to see whether we can find that sweet spot, you know, which might then generalize into a best dose for rifampicin.
Yeah, I could add, so within VQuim, we did do some PK work. For example, Andrew Burke from UQ led that, and it was important. It showed that the dosage that we had given was the right dose that achieved what should be therapeutic concentrations. And I think it would have been important if we had a negative trial that didn't show any difference ultimately, to explain why, because we wouldn't want to have underdosed. And I just, whilst we're talking about adding on studies, I'd also put in a plug for adding on studies that collect data about patient tolerability and preferences as well. And so, both in our study and also in TB Champ, we asked people what they thought about the drugs, how they found they tolerated it. And in our newer studies, we're going to do qualitative research as well, because often when you're developing guidelines, say through WHO, the considerations are not just around the point estimate of effect, but also around patient preferences as well. So it's really important to include those.
And I can see Debbie's actually got her hand up. So Debbie, yeah, you want to comment on the TDM?
Thanks, Gail. Look, I just want to remind people that ARC Diagnostics, with whom I have no association, no financial interest or whatever, make an immunoassay for linezolid, and that can easily be added on to something like the Abbott or other systems in the main biochemistry lab. So for MDR-TB, to actually be able to measure linezolid now in this setting is not difficult. The assays are not that expensive, and I'm sure that ARC would be open to discussions. They do have representatives throughout Asia. They would be open to discussions about actually using their assays. So it is able to be used in a routine biochemistry laboratory on many of the platforms that they would currently use. That's the only one that's available that's actually got TGA approval in Australia. There are Chinese and Indian companies who make immunoassays, but the only one with TGA approval is linezolid through ARC Diagnostics.
Thanks, Debbie. That's a fantastic tip. I'm going to go to a question that we received in our pre-talk registration. This is from Alison Ratcliffe at Royal Hobart Hospital in Tassie. She asks, of all three speakers, following your trials, what opportunities do you make available for researchers involved in these trials living in the countries where they are undertaken to present their findings at international conferences and meetings? I think this is a really important question. Perhaps we could, Nick, go ahead.
Sure. Yeah, it's really, really important. You know, these trials can't be done without really hard work at the trial sites, and there are many often it's the junior doctors, you know, the people with senior have got many things on their plate. Ultimately, the work is done by junior doctors, and it's a great learning experience to be involved in a clinical trial in terms of following a rigorous protocol, often get experience with use of new drugs, but it shouldn't stop there. There should be opportunities to engage academically when the results come out, and that's really important. There's, you know, the tension is that the trial also has to deliver publications promptly and so on, and very often, you know, people will say, I'll write stuff or I'll do this, but unsupported, nothing comes out. So what we're trying to do, you know, we tried many things, but certainly presentations at conferences, yes, we always, so for example, the one we're presenting most of the TRUNCATE-TB data is the union meeting. We've had 2 out of 4 speakers at our symposium, and each of the years have been regional speakers. So just a quick plug, if you're at the union, this year in Bali. We've got a symposium on biomarkers on the Saturday. Be very interesting data, but one presenter, the proteomics, will be presented by one of the junior investigators from the Philippines, and our artificial intelligence chest X-ray analysis will be presented by one of the investigators from Indonesia. So I think that's important. In terms of publications, what we're trying to do is set up a, hasn't started yet, but we've identified people, set up a mentoring process for writing papers. So 2 people from the regional sites, junior staff, selected on the basis of career, path, need, interest, to be co-leads on writing paper, 1 local senior mentor, and then myself and the trial team. So the plan is that we would hold staged teleconferences to discuss, initially, okay, we'll do the analysis and results. So the potential tables will be on the first call, and then have a discussion about what the take-home message is. People go off allocated bits of that to write the discussion, the methods, come back a month later, we see where we're at, and so on, all the way through to the process of submission, responding to reviewers' comments, proofreading that. So we'll see how that works out. I'm a little bit optimistic that it's got to be better than what we've done before, but we'll see.
Well, perhaps at the next one of these seminars, you can report back and let us know about, feeding back on how successful and the challenges with that. Maria and Greg, I don't know if you wanted to comment on that.
So as I mentioned earlier, one of the challenges of engaging young doctors in the site, in the clinical research site, is to ensure that they have a career in research to continue to engage them, or else you'll lose them to other endeavors, no?
Yeah.
So what we did was we collaborated with the Emory University with their track, the TB Research Advancement Center, where they have a mentoring program. So it's they're partnered with one international and me or my partner to come up with do grant writing as well as publication. And they also participate in Smart for TB. It's another consortium, has a phased program for young investigators. So we've enrolled all of them in that program. So where they already are writing, and we just got an award for TB Reach for a grant, so they're also very involved in that. So we make them be involved in grant writing and publications, so they will also develop their resume to become investigators themselves.
Please. Good blue adult.
Yep, and just I guess from our side of things, the time in terms of international presentations, usually the majority of opportunities are with sub-studies, and so we often try and give people opportunities to present those sub-study data at, I guess, the union meeting or regional meetings. And in Vietnam, to have a combination of people presenting the work to the Vietnamese audience as well. I think one other thing we're trying to do now more of is have PhD students from Vietnam involved in the trials throughout as well. So we have 2 current Vietnamese PhD students, and when they take on that work, then obviously they will lead particular components of the projects, and then that also builds their capacity too. So I agree with Nick, we should be doing as much as we can.
While I encourage people to ask questions, turn their mic on and ask them, I have another one perhaps I could ask of you, Greg. You mentioned that it would be important, rather than reinventing the wheel, is really to joining a clinical consortia that already exists or considering it. I don't know if you have any practical tips on how a site or an investigator, many of whom are gathered here today, would be able to actually, how do you begin that process? My experience is that you really do a clinical trial in a particular region, and then somehow as a consequence of conducting that clinical trial, you are then, you get the opportunity, I guess, to be introduced to a clinical trial network. So what are your practical tips for doing that?
It's a chicken or egg question, and it depends on who you are and where you are as well. So for us in Vietnam, the studies that we've done, I guess for almost 10 years, were investigator-initiated studies that were funded by an HMRC or equivalent. And once we had a track record, then we could present that track record to some of those trials networks. And that's certainly one way of doing it. These networks tend to have what they call recompetition cycles. So in the case of ACTG, it's every 7 years. In case of TB Trials Consortium, it's every 10 years. And they ask at that point, you have to sort of submit your CV basically as a group for your capacity. So yeah, so obviously that then will be stronger if you have existing trials experience. And some groups like ACTG have the capacity to have just one protocol specific trial site. And then if you do well at that, as Dr. Gler talked about before, then you maybe have more opportunities as well. But, you know, you can also talk to people who are already in the trials networks and just get involved in those sites as well, and see what opportunities there are.
Yeah, like just to add, for our site, we built it from the ground up. It was just from networking. One of my networks told me that there was an opening for a protocol-specific site, and you just apply. I applied, and we were accepted. And so we had to build. So it took us 2 years to be able to initiate enrollment, because ACTG has very stringent standards. So yeah, so it took us 2 years to build our capacity. And when you're in, it's a good thing to go in, because when you're in, it's also, it opens up doors.
Catherine Berry. Oh, sorry, go ahead.
Sorry, no, just to add, if people are interested, I'd say now is a particularly good time, given the money that's flooding into TB research. There is a shortage of trials capacity, because many the clinical trials, many networks have suddenly got money and a full portfolio of trials, and many of us are going to the same clinical site. So if if there's somebody on the call who does work or is at a site where you've got good capacity potentially to conduct clinical trials, with good lab and substantial TB burden, it's worth reaching out to any of the networks and say, how about it? It's not often the case, but now is a good time.
We have a couple of other questions, which I think Catherine Berry has asked before we wrap up, because I recognized it's nearly 2 hours in. What is the one thing we should advocate for to speed up TB clinical trials? I think we talked a lot about the challenges, particularly with getting, well, firstly, getting funding to the ideas that you want to, and then also really trying to get through all the red tape of governance bodies, etc., and how much they delay you. But what is the one thing we should all be advocating for to speed up TB clinical trials?
Hard to reduce it down to a short list, let alone one. But look, I think asking good questions is probably the first principle, because there's a lot of trials that are being done at the moment. And I think, study 31 is a good example of a study where there was a huge investment. The trial was done exceptionally well, but it has not really had a very great impact. And so I think we need to be thinking about how we can be efficient in our trials so that we choose questions that will actually change practice. And that doesn't just mean drug trials. It may also mean, other types of interventions as well.
Yeah, I think it's very hard to distill it down into one thing. Though everybody moans about ethics and IRBs, I personally don't think that's the issue. I think you need a robust process there, and part of the problem is that people submitting to ethics and IRBs don't have their act together at the point they submit, and that often delays things. Not always, of course, people are experienced, but you know, thinking these things through very carefully. So I don't think, you know, I think we need robust ethics review processes, and they need to go through this thing. And often, you know, you get good comments back from that. Yeah, I think it's very hard. I like Greg's point. Yes, choosing the right question and making sure it's durable for 5 to 7 years, because that's how long often it takes to get an answer. So choosing a question that's still going to be relevant 5 to 7 years down the line is really important. Other than that, speeding up, again, it's come up, talking to each other around the networks, you know, working with, knowing what other networks are doing, which sites they're working with, sharing notes about which sites, you know, what the strengths and weaknesses are, staggering enrollment, so that we can together have an efficient global network, which is partitioned into individual trials and individual networks, but still cooperates together, may help in that regard.
From my point of view, it's increasing clinical trial capacity in high burden countries, because like we're a very high burden country. We're only one site management organization that has the capacity to enroll right away, because other sites would still build their staffing as a non-established clinical trial unit. So there has to be an effort to increase trial capacity in high burden countries.
One of the things I think is, you know, this is my own perspective, and I'm not a pediatrician, but I do think it's important to address the vulnerable populations. And I was just wondering if Steve Graham, you'd like to make a comment on the inclusion of children and talking about, you know, formulations and age cutoffs with regards to drugs, please.
Sure, Gail. Yeah, I think, thank you for the opportunity. I saw that there was some questions came through around that. I think for TB now, we define child as being less than 10 and adolescents as 10 to 19. And in some ways that makes it a lot, that's an improvement really on this child being less than 15. We all understand that a certain age, particularly school age adolescents, there's a huge spectrum of cognitive capacity, development, social maturity, etc. But it also makes sense to cut off under 10 because the pattern of disease changes to much less bacteriological conformation, different radiological clinical presentation compared to the adolescents. So I think that hopefully will be a more convenient cutoff. Similarly, with the use of the child-friendly formulations for the younger weights under 25 kilos. And as we know, there's more and more child-friendly formulations, both first-line and second-line drugs now, which can be used in these trials. And we, I guess, sometimes make assumptions that these are better, but Graham, who I think's been on the line, has done some work as well on this in South Africa. But I think that's incredibly useful because these formulations are appropriately dosed. They're hopefully more child friendly. They don't require refrigeration, etc., etc. So that's a step forward. I wanted to just quickly mention that, as we all know, the major challenge for including children is the fact that so few of them are bacteriologically confirmed, and so then you are working with imperfect clinical standardized definitions for trying to categorize the cases, and that is I think the main challenge. But there has at least been now some success in child TB. We've had multi-country studies, both Africa and Asia involved in the same study. There's been the SHINE trial, MRC, TB Speed, UNITE funded, RAPID, EDCTP, all multi-center studies that have been quite successful in recent times. And one of the reasons that drives that is to get the large enough number of children who are bacteriologically confirmed often requires large numbers of clinical sites who have that capacity. I guess that's all I wanted to say. Thank you.
Thanks, Steve. I think we should, I'm just conscious of the time. We have 5 minutes before I'd like to wrap up. I should, Dr. We might, Dr. Ghaffar, would you like to perhaps put your question in the chat? I think Gail just, Dr. Matthews had a question that she wanted to raise.
Oh, I was, yes, I mean, I was going to ask something, it may be too big a topic or it may not, and it might show my ignorance of the TB literature. Social determinants of health, are they routinely collected in TB trials, and presumably food stress and work stress, etc., are major determinants of outcomes, and are they being collected, and should they be collected?
So I think certainly nutrition is identified as an important risk factor at a population level for developing TB. The challenge in trials which are not directly involving a nutritional intervention is the complexity of adding on too many questions about risk factors at the time that you're enrolling people. And so it would really depend upon the intervention that you're doing as to the extent to which it would be relevant to ask about nutrition. You'd be aware of the rations trial, which was a cluster randomized trial in India, which showed a substantial benefit of nutritional support. And so I think there's a lot more interest now in interventions that might involve nutrition. But you would expect, obviously, in randomized trials that the nutritional determinants would be equally balanced between groups. And so you may not adjust for them in the statistical analysis, but it might be a part of additional work looking at other modifiable risk factors in the same population. I don't know if that answers your question.
Yes, kind of, and probably it wasn't very clear. What I actually sort of meant more was like insecurity, so access to food, education, housing, you know, those kind of what we, I guess, would call social terms of health, that are not directly disease related, would probably affect your disease outcome. But it is another big topic, I guess. But nutrition.
Yeah, I mean, I just add to that. So Gail, the answer is not routinely collected, but often trials, if there's a health economic component to the trial, often those things, when you start to drill down to the questions, you know, when you get the health economists and social scientists involved, often these things are raised up. So I can think of several trials where this information is being or has been collected, but it's not routine.
I also just
one of the things it's very hard to do is it's very hard to do it on a global scale, right? Because it's very hard to then do it across countries when you're trying to measure these kind of stresses. So that was partly why I was interested. Sorry.
There's a body of work actually in Peru, led by a group from the UK. I'm very bad at names; they don't immediately come to mind. But Tom Wingfield, I think, is one of the investigators. They've done lovely work in terms of what constitutes treatment support, and it's not just, you know, it's not just one thing. It's not about observed therapy. It is really about supporting a person and the people within their family to get through the course of TB treatment. And they have shown, you know, huge separations in your Cox proportional hazards curves in terms of the benefit that treatment support package, which constitutes everything from, you know, education, social welfare, the lot, the differences it can make are huge. And it really speaks to whether we really need better drugs, which, you know, I'm a big proponent of. I do think that the drugs we've got are terrible and we do need to improve them. But really, we do know that TB is a disease of poverty. And if we eliminated poverty, we'd get rid of TB, but that's not going to happen. Dr. Ghaffar, thank you for waiting.
Attention, please.
Okay, thank you very much for the opportunity. I'm Fajri from McGill in Canada. So I'd just like to ask like Greg or Nick, your experiences and challenges in finding new sites for clinical trials, and how did you do capacity building for these new sites? Because I have a secondary affiliation at the University of Pajajaran in Bandung, Indonesia, where we have a good capacity to do clinical trials, but we are currently, you know, being approached by so many consortiums, but our sites currently is in we call like the saturation levels. It's really hard to recruit participants in our sites. And when we offer to include new sites in different provinces to this new consortium, to this trial consortium, they tend to, to be hesitant and refuse to include these new sites. And that's and they tend to be hesitant to do a capacity building totally from the beginning in this new site. So I'd like to ask your experiences and like challenges in doing capacity building in new, completely new trial sites.
Sorry, I don't know whether that was to me or to Greg.
It can be to you and to Greg, because for Greg, I noticed that there are so many trial sites in Vietnam involved in your trials as well, and that makes like multi-country, multi-site trials. So, yeah.
Greg, do you want to go first?
Oh, just yes, in the short time we have available. Thanks, Fajri, for your question. I think there needs to be a depth of experience depending upon what the activity is that's needed. So for the coordinating center for the country, there needs to be a high level of clinical trials capacity. So so at the Clinical Trials Unit in the Sydney Vietnam Institute, for example, the team there, are quite familiar with some of the design aspects of trials and the capacity to be able to oversee the trials well. And then at sites, to make sure that the site PIs there have the capacity that they require, clinical capacity and GCP training, and then training the specific protocol of the study. And so I think it's a constant effort to always be learning ourselves, but also whenever there's a new staff member that arrives in a site, to bring them up to capacity by teaching, by mentoring, by in-trial supervision, which I think is probably the way people learn the most. That is getting feedback on how they do in the trial, and then that helps to build their capacity.
And I would say, in terms of, just very briefly, in terms of going to new sites, selecting new sites, there are two things that I look for. Some people come with shopping lists of 10 pages, they must have this, must have this. I only look for two things. One, is there a PI that's engaged and has got time and really enthusiastic, and you can tell will make this happen? And number two, is there a lab, microbiology lab, because it's so essential for drug trials. Your TB lab needs to deliver reliable midget results. If you've got those two things, pretty much everything else can be made to work, in my experience. Not always. There's always a bit of a revolving door. You try new sites and some don't work, some do, but those would be the two essential things. If you haven't got a leadership from the top, and you haven't got the micro lab capacity robust that can support it with reliable data, then I think it's very difficult.
Okay, I think we have reached just after 3 p.m, so it's probably a good time to wrap up. I think that this was a really successful seminar. I'm very grateful to all three speakers for taking 2 hours out of incredibly busy schedules to do this. I hope that this will lead to better questions for clinical trials and participation in those clinical trials from young investigators like myself, but also clinical trial sites within the region. I'd love to see, Greg does incredible work in clinical trials in Australia, and I'd love to see more of it done in Australia for selfish reasons. But I'm very, very heartened by what we've heard today, and what I think that can be accomplished for TB. And it's testament to the fact that we had, over 100 people register for this. And we had 75 people attend today, and it was from, throughout Asia Pacific and beyond. I think that just really speaks to the fact that, as Greg said, we are at the epicenter of a big problem that deserves addressing. And I won't get on my high horse about how research in other fields, there is certainly a bias, but I really want to thank everyone today for attending, for their contribution. I will summarize this session from all three speakers and send it out to those who registered. It will be available on the ACTnet website for people to view at their own pleasure once we've edited edited the bits that don't matter out. And thank you once again. And, you know, any final comments, Gail Matthews?
No, it's been wonderfully informative for me. I'm not coming from the TB world, so my thanks as well to 3 great speakers and all the comments, questions.
Bye, everyone.